Have you ever wondered why your brain is wrinkly? Are wrinkles associated with intelligence? How do wrinkles develop? This video will serve to dispel these mysteries surrounding the wrinkles on your brain and explain differences between individuals and organisms. Firstly, how do these wrinkles develop? Scientists have figured out ways to use a gel model to replicate the folding process in real brains. As we may observe from this clip, the gel brains absorb solvent that causes the outer layer to swell, resulting in folds that we find on human brains. This process is called differential swelling, when certain parts of the brain grow faster than others. More specifically, the gray matter or outer layer expands faster than the white matter or inner layer. In humans, the cortex expansion begins to occur starting from the uterus at about 20 weeks and continues until age 1 to 2. This process can also be called gyrification, the process of folding gyri, bumps, or sulci, divots. When non-dividing neurons crowd up the brain surface, the brain gets closer to reaching critical density, then begins to wrinkle. This type of folding occurs to optimize the amount of brain mass that could fit in our heads and increase surface area of the cerebral cortex. As quoted from the Smithsonian Magazine, if you look at a human brain, you only see about a third of its surface. The other 2/3 are hidden in its folds. If you could spread it out flat on a table, it would be 2,500 square centimeters, or the size of a small tablecloth. Thus, more wrinkly brains mean that the brain has a larger surface area and thus a higher brain volume ratio. Wrinkles also shorten the distance that axons and white matter have to travel to communicate between regions, which maximizes efficiency. Does this mean that wrinkling is truly associated with intelligence? Research has shown that wrinkles generally reflect intelligence. In fact, the most correlation has been found between intelligence and folding in the temporal-occipital lobe. Brain waves, or connections between neurons, can also be predicted by cerebral cortex outer folds. A higher concentration of folding does relate to more cognitive complexity. For example, the frontal cortex is typically more wrinkly than the back because it performs more complex actions and abstract thinking. Differences have also been discovered between genders. Female brains demonstrated a more positive relationship between degree of brain folding and intelligence level. However, no significant negative relationships were observed in either male or female brains, so there was an overall positive relationship between folding and intelligence. Now, what do these wrinkles really look like? Primary fold locations are generally consistent between individuals. These include the central sulcus, which divides the frontal and parietal lobes, the Sylvian fissure, which divides off the temporal lobe, lateral fissure, and longitudinal fissure, which goes down the middle of the brain. Secondary and tertiary folds can be more random across individuals. Wrinkles also vary greatly between different species. Organisms with larger, wrinklier brains include primates, dolphins, humans, elephants, chimpanzees, and pigs. Humans have the most wrinkles, which means that they're the most evolutionarily advanced. Organisms with smaller, smoother brains include mice, manatees, rats, and squirrels. Smoother brains in humans have often been associated with neurological impairment. For example, lissencephaly is a condition where the brain lacks its normal bumps and folds. This condition occurs when the outer brain regions don't expand properly due to mutations that cause impaired neuronal migration. In other words, new cells don't travel to the brain surface during gestation, and not enough cell layers are formed. Symptoms include intellectual disabilities, muscle spasms, and a shortened lifespan. Polymicrogyria is another condition where the brain contains an excessive amount of tightly packed ridges, or gyri. Symptoms include seizures, disabilities, neurodevelopmental delay, autism, and schizophrenia. Not having major brain folds in the right places can also cause neurological dysfunction. How have scientists been able to arrive at these conclusions regarding brain wrinkles? A type of technology called DTI sensing, or diffusion tensor imaging, allows researchers to look inside the brain at neuron communication. It is similar to MRI imaging, which measures the concentration of signals sent throughout the brain. Scientists have also investigated brain organoids that are grown on a microchip, which simulate a real brain at an early age. They mirror real brains in structure, function, and nutrient delivery. In conclusion, there is sufficient evidence proving that more intelligent organisms do in fact have wrinklier brains. Thank you.